We are happy to be joined today by Denise Galloway, head of the Cancer Biology Program at Fred Hutchinson Cancer Research Center, which shares the 5th annual AACR Team Science Award with the University of Washington for their 20-year collaboration on research on the human papillomavirus. Thank you so much for being here, and congratulations.
Thank you very much for having me.
Your work on the HPV resulted in a tremendous increase in our understanding of the human papillomavirus and a vaccine which is now in use around the world. What part of that research was the turning point when you knew that you were onto something?
I think there were two turning points. In the early days, in the early 80s, when HPVs were first found in cervical cancers, DNA testing for HPV was not very good, and the results were very inconsistent. Some studies saw HPV in only the cancers, some saw it in normal tissues, some saw it in other than genital tissues, and it was really unclear whether this was going to be interesting or not. And as DNA testing improved, all of a sudden, all of the studies began to be consistent, including ours, that we saw HPV DNA in almost all cervical cancers, the majority of other anogenital cancers, and very little in normal genital tracts of age-matched women. You did see some in young women, but it wasn't integrated. And the story became very consistent, and it was clear that it seemed to be associated with the cancers and not with normal tissue. So that was very exciting. You knew it was going to be real. I think the second turning point came when it was possible to make virus-like particles. Again, several labs right around the early 90s showed that if you took the major capsid protein from any one of several HPV types, that it would fold into something that looked to be identical to real virus, that was immunogenic when put into animals, and was likely going to be the basis for a prophylactic vaccine. And I think that was a very exciting time too.
Today we hear a lot about different institutions collaborating and sharing resources and knowledge for the sake of research. 20 years ago this wasn't necessarily the case. How did your partnership with the University of Washington get started?
Well, first the partnership, the interdisciplinary part of this began within the Hutchinson Center. So there are different divisions that don't always know what the other division is doing. And at a retreat, a center-wide retreat, our group that was working on basic molecular biology of viruses with Jim McDougall ran into Janet Daling, who was an epidemiologist in a different building that we didn't normally interact with. And she was working on cervical cancer too, doing case control studies, population-based case control studies. And we realized that if we put the molecular biology together with the epidemiology, that would be something exciting and new. And so once that got started, doing these surveys looking at cancers, we realized that we didn't really know much about the infection. Who got infected? How long did infection persist? Was it prevalent? All of those sorts of questions, which we call the natural history of HPV infection, were unknown. And at the University of Washington, there was an outstanding group that did infectious disease, sexually transmitted infections as really a strength at the University of Washington. And they were beginning to be interested in the same types of questions for HPV. And so again, we thought if we brought our resources together, we could now start answering questions not only about cancer, but about the infection itself and who had it and then who was likely to go on to progress to cancer.
Much of your research includes very sensitive and personal information. How did you gather and validate this information?
Well, luckily that happens to be what the epidemiologists do, not what I do. But they are really very skilled at asking those sorts of very delicate questions. How many partners have you had? What types of sexual activity do you have, et cetera? And there are really two different approaches that these different groups have taken. For the population-based studies, they have very skilled interviewers who are used to going out and asking these questions. They often will show pictures or offer choices. So, for example, if you said, how many partners have you had? You don't have to say, well, let me see, 1, 2, 3, 2. They may offer you a choice, 0 to 5, 5 to 10, 10 to 20. They keep their eyes down so there's no type of value judgment when you answer. All of the information that they get is kept very confidentially. The interviewers have to sign confidentiality agreements that they will not disclose anything. And then once it's entered into the databases, almost no one sees who is associated with that data. So for example, when the laboratory gets it, we get coded samples where there's no identification of the patients or any of the information that's associated with them. Now, in the natural history studies, we were dealing primarily with young college students, and they have a completely different way of interacting. They filled out personal diaries, they filled out internet-associated diaries that had a very secure website, and so there was less one-on-one interaction with them because they were very comfortable doing all of the electronic entering of their own data.
What are some of the other strategies for early detection and treatment of HPV-related cancers?
Well, early detection relies primarily on cytology to detect the precursors, but I think there's an increasing shift to using DNA testing as really an important adjunct to that, or I think in time will supplant that. We know which are the types that are high risk, and you can ask whether or not someone has high-risk DNA, and that puts them in a very different risk category compared to having only low-risk HPV types or no HPV. And the algorithms then will change to follow people depending on their HPV type. So for early detection, that's still the most likely or the best approach to take. I should say that there's a lot of interest also in oropharyngeal cancer, and the increasing finding that about at least half of tonsils and oropharynx cancers are associated with HPV. And there the big interest is to find out how do we do early detection? What sort of samples should we be taking? And it's really not known at this point. In terms of treatment, the best treatments are still LEAP, laser removal of the lesions. Early stage cancers are very, very treatable when they're still contained within the cervix. Following that, there's the addition of chemotherapy or radiation, but there's a lot of interest in developing immunotherapy to see if we can really help the surgical removal of these cancers.
So what's next for your research?
Well, there are still many areas for HPV that we don't understand all of the natural history. As I mentioned, the oropharynx is one. When do these infections occur? Are they at the same time that you get genital tract infections? We don't really know. How do they progress? That's an area of interest as well. We're also moving on to some other HPV types that are not genital tract associated, but are common infections of the skin. And there's a lot of interest in whether or not they play a role in squamous cell cancers of the skin. And we have a big study following a population of organ transplant recipients who are at extremely high risk of developing skin cancers. And we're following them for the development of skin cancer and to determine how their HPV behaves after organ transplantation when they're on these chronic immunosuppressive drugs. Another large interest of my lab is simply to ask whether or not other cancers have a viral etiology. And the reasoning behind that is that all of the cancers that we know that have viral etiologies increase disproportionately in immunosuppressed individuals. They are much higher levels than in the normal population. All cancers increase a little bit, but some of them are really high. And while there are anogenital cancers, Kaposi's sarcoma, lymphomas, things that we know have a viral origin, there are other cancers that increase also, like the squamous cell skin cancers, lung cancers, multiple myeloma, lymphomas that are not known to be EBV positive. All of these are great candidates for having a viral origin, but we don't know what it is. And so we're using some of the new high-tech, high throughput sequencing to see if we can find any of these new agents. And hopefully we could make the same strides in those cancers that we have with HPV.
Well, congratulations again, and thank you so much for being here.
Well, thank you very much for having me. It's been a pleasure.